Welcome back to the Molpigs podcast. Today our guest is Grigory Tikhomirov. Also here today are Boya,
Hello.
Eric,
Hi.
and I'm Hannah. Greg's an assistant professor in electrical engineering and computer sciences with a background in chemistry, bioengineering, medicine, and nanotechnology. He has recently started the T-Lab at Berkeley to pursue the design and fabrication of devices with atomic precision by combining the strengths of rational top-down engineering and bottom-up biomolecular assembly. A key goal is to adopt the powerful but still proof-of-concept self-assembly approaches of DNA nanotechnology to engineer new, useful devices. Greg, hi.
Oh, hello. Thanks so much for inviting me here. It's an honor to be on a podcast and talk about my favorite topic, molecular programming.
Yeah, I really look forward to talking to you today. You started out more in chemistry. How did you find out about and move into molecular programming?
Well, I feel like I have always been a molecular programmer, but what really got me into the field is seeing Paul Rodemun's smiley face DNA origami paper, and as a chemist, I really like to synthesize large, beautiful molecules. And when I saw this giant molecule, I couldn't believe my eyes. It's like it's amazing. And ever since then, I thought that I need to join this field. And I had a dream to reprogram molecules to build new life forms, because I was in maybe middle school or early high school. I kind of encountered first time, death and sickness in the family, and I thought, why do people have to get sick? Why do people have to die? And can we kind of re-engineer people using kind of more rational principles, like principles of engineering, where you can use transistor, resistor, and capacitor and build very complex circuits, and you understand actually what each module is doing. And I thought, okay, I can use supramolecular chemistry to do that, because you cannot really synthesize a cell, but you can self-assemble a cell using non-covalent interactions such as hydrogen bonds, pi-pi stacking. And I tried to design molecules that can self-assemble into complex structures, but it's very tedious to try to use organic chemistry, because you have to do a lot of synthesis, and you carry out this multi-step synthesis. At the end of the 20 steps, starting with 100 gram, you get 1 milligram of compound that maybe self-assembles, maybe doesn't. And you spend 1 year synthesizing and then looking what you got, and it doesn't work. It works, but it makes very, very simple structures. Like I self-assembled nanotubes, not carbon nanotubes, but organic tubes that assemble from 6 rosettes, and each rosette is made of guanine and cytosine molecule linked by 60-degree angle. So kind of we borrowed from DNA, but they're still too simple. And I realized that this is not sustainable. And I kind of got frustrated, and I realized that my PhD is kind of a failure, and I need to join different field. So I went into cancer research. And you can say it's some kind of molecular programming too. So we try to build activatable nanoparticles and molecules that detect and treat cancer based on the presence of certain enzymes that are associated with cancer. So versus traditional cancer therapy, where you deliver drug everywhere, and it kind of kills healthy cells too, but it kills more quickly dividing cancer cells. Instead of that, if you make it targeted, then we can kill cancer. And this was very interesting research. And what I learned is that chemist and bioengineer can make a huge difference in medical practice. Of course, it takes about 10 years to introduce a new drug to the market and go through all FDA approvals, and thousands of very beautiful ideas on smart activatable drugs fail because they just don't pass toxicity and don't get to preclinical stage and they fail in animals. And yeah, so at some point that I realized that I have to leave science and go to industry or try to find something that is really exciting me. And then I was lucky enough to join Lulu Chian's lab, and she's been absolutely amazing, his advisor. And that's kind of saved me from going from leaving academia, and got me again very excited about molecular programming. And I realized that is actually the way to build complex uh, architectures, like new life forms. And what I'm doing right now in the lab, that maybe not necessarily that far-fetched as building new life forms. It's more making devices that have molecular programming capabilities and intelligence of molecules. But yeah, that's kind of, sorry, that was a long explanation how I got into molecular programming.
Yeah, I think I can't speak for all of us, but definitely for me, the medical applications of molecular programming in the future really excite and drive me. Where would you say we are kind of today? Like, what would you say the timeline is, in your opinion, for all of these different milestones?
Yeah, I think we have a lot of exciting developments right now for biomedical applications, and in when it comes to using molecular programming to clinical purposes, I think the most exciting example for me was Sean Douglas, a paper from Sean Douglas and Ido and George Church on this clamshell robot that um, basically a simple molecular computation, an AND gate or an OR gate. You can program two locks to open two different molecular signals on the surface of the cells, and open up to expose this molecule that can trigger apoptosis, the cell death. And so for me, it still remains the most exciting demonstration so far. And that's a science paper in 2010. And I don't feel like we have anything more complex than that in the field. and and of course there is a lot of developments since then. People study, we need a lot of fundamental studies, what happens to DNA structures, how to make them more stable in biological media, and many groups around the world working on that. And there are a lot of fundamental studies that contribute. We still probably a decade away from, at least from clinically approved DNA nanotechnology. But I think it's one of the next big steps for DNA nanotech is to make huge difference in medical practice, because the capabilities and just friendliness of DNA and programming and just the right scale, because we can build now structures that can interface very well with the cell surface, and have enough complexity to beat any existing therapies, maybe except for T cells, but T cell CAR T cells therapy, but it's very expensive. It's still very hard. I think DNA nanotech will have a lot of applications very soon in biomedical practice.
So as far as those biomedical applications, what do you think are kind of the next steps we need to get from that 2010 Sean Douglas paper to the decade away FDA approval of a DNA nanotech-based delivery device? Like, is there anything else in the academic space that needs to be done to prove this concept? Or at this point, is it kind of somebody needs to figure out the large-scale commercial production of these structures and do the pharmacokinetics of it?
That's a good question, and I think it's both. Both. We need more fundamental studies in our community, and also more translational studies. And we need people to actually try and to work more on this kind of tedious translational stuff, maybe conceptually not so exciting, but and it's very expensive uh, because you need a lot of animal studies, maybe large animal studies. But we need someone who can take those constructs that the community developed and try to see how they behave in all these complex environments, and more carefully zoom in on specific type of diseases. And I know there are already several startups in that area, and they're working on this with private funding. Yeah, we're going there. I mean, both things that you mentioned are happening both in academia and kind of industry.
Is your lab trying to push molecular programming to the application side, or you're or to more try to commercialize those devices you build, or you're more focused on the basic science or proof of principle side?
We are working on both. We are trying to develop some new concepts, but also we are trying to work on something that is has a potential, at least, of being practical. But we not really like just want it to be practical. We want it to be conceptually interesting for us, and I mean, just we don't do something for the sake of being practical, but we try to select projects that combine being the properties of being fundamentally exciting, and at the same time can be translated to real life applications. Because I feel like a lot of science is very exciting, and we have enough ideas to also focus on something that give back to society, that whose tax money we use to do research. I mean, who wouldn't like to live a little bit longer and healthier, and use devices that compute much faster?
So on that note, one of the big reasons we invited you on the podcast today is because you're a relatively new PI who is very kind of prominent in the field. And so we wanted to hear your experience starting up a lab and starting to develop your own scientific questions. So can you first tell us a little bit about kind of what the focus of your lab is and what the big overarching vision is for you?
Yeah, sure. I'm far from prominent PI. I just started my lab in July last year, and I feel we're still building the lab. We're still waiting for shipment of some equipment, and it's been somewhat challenging to start it during pandemic, and also in a big university like Berkeley, where things don't get done as quickly as at some private schools like Caltech. uh, but yeah, the focus of my lab is try to take all these developments in molecular programming field and embed this kind of molecular intelligence into non DNA building blocks, because DNA is kind of boring from physics viewpoint. I mean, you can do a lot of interesting thermodynamics and biophysics, but it doesn't have conductivity of carbon nanotubes, so it doesn't have much optical properties to talk about. And so the one big direction is how do we take those building blocks with interesting physical characteristics, high performance building blocks like quantum dots, metals, semiconductors, and embed molecular recognition capabilities into them. And the reason we want to do that is we want for them to self-assemble into larger devices, complex devices, not just periodic crystals that many groups have demonstrated so far, but we want something that has thousands of uniquely addressable building blocks, something like SST tiles, so DNA origami. But imagine instead of each staple you have a different building block, and then not periodic and unbounded crystals like sodium chloride. There's something finite size, and you can precisely control the shape and physical properties. So we basically try to develop a new nanotechnology, not specifically focusing on any device, but we want to provide kind of general tool for community to use to do nanofabrication that is not possible with top-down fabrication, because many people think that DNA nanotech is going to replace semiconductor manufacturing and TSMC Intel factory. That's probably not going to happen. I'm quite sure it's not going to happen. So this lithography and top-down fabrication is probably the most sophisticated technology that humanity ever developed, and the precision with which and fidelity and low error rates that people can achieve in semiconductor manufacturing is not achievable with DNA nanotech. But DNA nanotech has some unique niche, and molecular programming in general, that traditional top-down fabrication cannot achieve, and we are focusing on a suite of those technologies that uniquely enabled by molecular programming. And like also, so I mentioned self-assembly into new nanotechnology, and also integrating this new technology with existing CMOS, so top-down lithography. And also we're interested in various, I would say, nanorobotics directions, where we try to combine also high-performance materials like magnetic particles for non-invasive control and imaging of the robots inside the body.
So when you do you envision some of these kind of interfacing with lithography? Is that what you were saying? And kind of what particular things could we do with this? Is this to build kind of specialized structures that kind of lithography doesn't do well with?
Yeah, so lithography is very good with working with silicon and CMOS compatible processes, something that you can carve out of silicon, or something you can deposit on the top of silicon, and you typically just spin a photoresist photosensitive layer, then you draw something with it with light or e-beam, and then you deposit something, strip photoresist, and you repeat this process many times until you get this structure. And so the materials that are compatible with this approach kind of limited, and ideally we would like to be able to, and also precision is somewhat limited, and scalability, because you have wafer scale. I mean, although now you can do many wafers per minute and state of the art tools. So we want to develop self-assembly in 3D, and we want to use dielectrics and something like that is transparent to the light, doesn't absorb light very much. And one concept that we're very interested in is inverse design. So I don't know if you've heard of inverse design. It's different in different fields, but there is inverse designs in photonics. They allow to have an arbitrary optical transformation of light. You can imagine it could be something like broadband light, so white light spectrum comes in, and you have this tiny micron scale. It should be about a couple wavelengths of light cube, and depending on dielectric distribution within this cube, we can have almost arbitrary transformation of light. For example, it could sort this light into RGB components, so that's so-called Bayer filter. So it's something that splits white light into red, green, and blue light. And we're all using it right now, and because with our color cameras, image sensors. And typical Bayer filters absorb 2/3 of the photons. So with inverse design, it's possible to build a Bayer filter that utilizes almost every single photon. So in theory, people show that you can get 95% color sorting without losing any photons. But no one can fabricate something like this for visible spectrum, because you actually need. So I mentioned dielectric constant distribution with precision of about 50, 60 nanometers. So you need 50, 60 nanometer building block that is, and you actually need just two of them to make an arbitrary function, so digital material. So there is also a concept that had been proposed about 5 years ago. So you need just two building blocks, but you need to precisely arrange them in this micron scale cube. And you need about, if you calculate, if it's 16 nanometers, it's about 5,000 building blocks. And but they have to be non-DNA. They have to be something like silicon and titanium dioxide, something that doesn't absorb the light. So you cannot use silicon that is compatible with CMOS. No one was able to build this, even though in theory it has been predicted a while ago. People built something like this in microwave, where you can use just a 3D printer and you can use polymer and air to different polymers with different refraction indices. But these are like centimeter scale stuff. You cannot really put it in as a pixel in your camera. It'll be a very big camera. And CMOS fabrication lithography cannot make anything like this. Potentially, you could use layer by layer fabrication, but again, it's not possible with currently materials compatible with lithography. So that's just one example, but you can also think about optical computing, because you can build a logic gate, optical logic gate, using the same inverse design approach. And you can do computation really, really fast and very energy efficiently with light compared to electrons. And you can make objects basically invisible, and you can have a lot of interesting functions. And this is very kind of computationally expensive process, because you basically solve it in a direct way. You just you have this function, and you optimize your voxel one by one, kind of gradient descent, and you solve in Maxwell equations for each of the distribution of the voxels in this volume. So we have somebody who is very good at this in the lab, and so Durkheim found a way to optimize the structures, but it takes forever, even on supercomputers. Now we develop in a way to embed information to those nanoscale voxels and self-assemble them, kind of by putting DNA suit on non-DNA voxels.
So it sounds kind of like it's a incredibly complicated process to go from, I want to make a optical computer or a optical transistor or any of these kind of metamaterial structures, to I need to find somebody who knows how to make the programs that solve these Maxwell equations. I need to find people who know DNA nanofabrications to position these things. How have you gone about like building that team? Let me rephrase that. How have you gone from the initial ideation of this is what I want to build to building the team that you're now attempting to build these structures with?
Yeah, that's a good question. And I guess my lab is very small, and I wanted to for this kind of interdisciplinary projects, I wanted someone who has different expertise from me. I couldn't afford to hire another person with DNA and nanotechnology expertise. So I am the one who kind of is a molecular programmer in our lab, even though I don't really program. But first few people I managed to attract are all from different fields. So first postdoc, very talented postdoc she got her PhD in MEMS. And so she's developing actually ways to integrate self-assembled structures with CMOS, and her whole PhD was just in clean room. She's extremely efficient with all this, proficient with the tools in nanofabrication, microfabrication. Also, another postdoc is purely chemist, so she's a specialist in nanoparticle synthesis. And another very talented undergrad. So we started working during pandemic, and I gave him project. Okay, make this wireframe, so type of a suit that we can put on DNA nanoparticles. And it turned out to be quite hard to design this wireframe suit. And he wrote the whole software, just in 3 months. And he keeps developing, and he's a wizard programmer. So everyone knows much more than me in the lab. And I kind of try to show them the beauty of molecular programming, maybe get excited about some molecular programming concepts, but everyone has a unique strengths and talent. And some people, they span different disciplines. Like Durham, he's designing inverse design, molecular dynamics, and artificial intelligence, everything. So I kind of got lucky with first few people who joined my lab, and they can embrace this collaborative environment where everyone is very good at something, and people talk to come up with new nanotechnologies. And also, for many grad students, I feel my philosophy is kind of try to get people who obsessed with something and very excited about some idea. And so this has a caveat that many times they don't want to work on my ideas, and this is totally fine. I'm just want to help them to maybe. There is a reason why they join my lab, because they see that potentially molecular programming and self-assembly can help them achieve their dream. So we have the very first graduate student joined my lab. So Benjamin, he's a MEMS person. He got his undergraduate and master's in electrical engineering, computer science, and at MIT, and he's interested in brain-machine interface because he played hockey, and he got brain injury, and so since then he wanted to kind of wait to debug electronic circuits of the brain. And he realized that with existing approaches, we cannot really scalably address every neuron. We cannot even address 100 neurons in a specific way, because you typically have to drill a hole in the skull and insert an electrode to stimulate neurons. So he thought maybe nanorobotics, molecular programming, DNA nanotech can give a tool. And so he's now working on a very exciting project that he came up by himself completely. And I'm just, helping in any way I can. But, he's totally self-driven. And, I'm very lucky to have people like this joining my lab, not necessarily buying my agenda.
So how did you get to this point? Like back when you were a postdoc, did you just wake up one morning and like, I'm ready to start a lab now? And then, or how did that happen? And when you did feel that you were ready, what was the process from the idea of like a lab to where you are now?
I always thought I wanted to be a faculty, and because I really enjoy doing research, I would say probably in high school I realized that I want to be a scientist. I was just not. I spent a lot of time in my postdoc, much longer than typically people spend. I did 3 postdocs in 3 totally different areas, and also very different from my PhD, looking for the ways to build artificial life, and also at the same time making an impact on society. And as I mentioned, being in Lulu Chan's lab was transformational for me, because I kind of finally was able to acquire the skills and also learn a lot from Lulu on how to start a lab, because I was the first person who joined her lab. And yeah, I mean, I've been on a job market for 3 years. It took me a while to find this position, and yeah, I just could not imagine myself doing something else than I like teaching. I realized actually that I like writing grants, although I would like to spend a bit more time with people in the lab, because in the first year you have to spend a lot of time writing grants. uh, but yeah, I really enjoy I, academic life. I didn't realize that I could. I was not sure if I would be successful, but now I kind of got a couple of grants, and I don't know if I can write papers. So we'll find out.
Congratulations on the grants. I'm wondering, do you have any plans about how you spend your grant money? Like you started your lab and you need to purchase a lot of stuff. And how was your decision-making process regarding what to purchase and what not?
Yeah, when you write grants, typically you can hire people. Yeah, so I'm investing in people. So all the grants go to fund people, and I try to use my startup funds for materials, and I had to spend much more than I expected for lab renovation, and so I need to keep raising more money because two new grad students come in joining the lab in September, and they're both international, and international students cost a lot, uh, in Berkeley at least. Yeah, but it's just almost everything goes into funding people, and I think this is the most important um, investment. But we also realized that one way to simplify life of graduate students and postdocs is to automate some processes like tedious pipetting. So the very first instrument that I bought is a mixing robot, those echo ultrasonic mixing robots. And for me, I spend a lot of time being a robot in a lab, and it's this Mona Lisa. It took me and Philip, who was a grad student when I was in Lulu's lab, 1 week to mix, and we made some mistakes because it's like 10,000 of strands you have to pipette, 64 tiles, mix them again, and now a mixing robot can do this in 30 minutes with almost no mistakes. I mean, no really mistakes. And so yeah, I didn't want my students to struggle through that being robots, and I got this. This is my big first splurge.
How would you say that having that mixing robot has changed the way you approach research without having to do the kind of the robotic parts by hand? How has that changed the way that you guys can approach problems?
It totally changed our life and let us try many more ideas. And that's why I like molecular programming too, because it almost always works. With and it takes just a couple of days for a new design iteration. Sometimes less. You can anneal your origami, look at it in the evening, and by FM or TEM. And as I mentioned, when I was doing it with covalent chemistry, yeah, you spend a year synthesizing a molecule, and if you're lucky enough to get it in enough quantity to study it by different techniques, it still doesn't form a complicated structure. But with molecular programming, you can program really, really complex structure, and if you pay attention to kind of very reasonable design rules, it works. And you can test out many designs, whatever you're capable of, AFM, looking by various other techniques like fluorescence.
So you spent a lot of your first year working on grants. Are you finding that time is opening up and you're able to do more kind of research, or in your position, are you finding yourself mostly at kind of the admin level, kind of writing grants and looking after your lab?
uh, Yeah, I'm still writing grants. I still need to get more money. I mean, we can kind of breathe a little bit more, but yeah, I just have so many ideas. I feel like very exciting. So we need to hire a few more people, and yeah, and it's besides grant writing, it's also a lot of dealing with administrative things. uh, like serving on committee, and also teaching takes a lot of time, especially because I'm kind of biologist and chemist by background, but I am now in engineering department. So and I had to teach I I was teaching for the first time in this semester. I was teaching nanofabrication, and I think most of the students knew much more than me at the beginning of the class. I think now we know about the same amount, but somehow I managed to teach this class, and it took a lot of time preparing the lectures.
Did you expect that this would be the way your time was divided, or have there been any things which you were surprised by when going into this position?
Yeah, I suspected that I would spend less time doing science, and I still want to go to the lab. I mean, yeah, yesterday I went to the lab to make some couple of origamis, just because everyone is so busy with their project. I cannot really ask anyone to try my new pet idea, so I just did it myself. But I was hoping that I spend more time talking to my students, but especially because everyone is from different background. But yeah, and I'm still waiting for some rich person coming and donating us a lot of money so I can do more science.
Do you have any advice for anyone who's kind of who's starting out doing the same thing?
Yeah, yeah. If you let's say join in molecular programming field and you feel curious about it, but uh, you think you're not prepared enough, let me tell you my experience when I joined Lulu Chen's group at Caltech. I felt like the most stupid person in a group. Everyone is proving computability theorems and doing some crazy origami design, and I don't understand anything. I didn't understand 90% of the things people talked about group meetings, because we had this joint meeting with Eric Winfrey and Paul Rodemund labs. And I'm just a chemist, yeah, I can synthesize this molecule, but this is absolutely useless knowledge. And so the learning curve was very steep, and I felt like initially for first year, I don't belong. Everyone is way smarter. And then I'm like molecular programmer. I cannot even program in Python. I mean, I can write some simple code, but nothing like people can do in this field. And yeah, it took some time to realize that there is a niche for everyone, and so don't feel intimidated if you want to join a field that looks very interesting to you, but you're scared. I mean, also it's important. Yeah. Talking about advice, I think it's very important to find a good advisor. But also realize that it's no matter what advisor you have, you can succeed. I mean, as I mentioned, I was very fortunate that Lulu took me on as a first person in her lab, and I learned a great deal from her, and I feel she's the most amazing advisor and scientist and person. And so one advice I can tell you: join Lulu's lab. But there are many other great PIs. And what other advice? Yeah, I feel like there is a place and space for everyone, whether you're physicist, chemist, engineer, in molecular programming field, or in any field.
Kind of on that same note, how would you say your perspective has changed as you've switched from advisee to advisor and as postdoc to PI, especially how it's kind of changed your interactions with the rest of the field now that it's PI to PI and you're interacting a lot more with the funding agencies themselves?
Interesting. Yeah, I feel like when I was a postdoc, a grad student, I was much more aggressive in criticizing other people, and if I feel like that it's somewhat not logical what they propose, it's somewhat dishonest, I call them out. But now I am much more timid. So I don't know what someone said, kick up and kiss down. So I'm like with people in my lab. I'm trying to be as supportive as possible. But people who hire me in hierarchy, I try to challenge them. And yeah, it's been now a PI. I try to kind of instill agency in everyone. I am far from hierarchical. I mean, everyone has their own project. I don't have hierarchy in a lab. A grad student, an undergraduate student, and postdoc, and me, we are kind of on the same level. Although it's, of course, not possible to completely create this. But yeah, everyone has their own project, and that I hope they like, and I try to instill the agency in people, feel like, well, they actually driving the project, and I'm just helping them to succeed. And at the same time, we try to, if I it was my will, I would work on very few papers and some transformational research. But since grad students and everybody else need publications to advance their careers, so it really changed my way to to guide those projects. If I'm the only one working on this project, okay, that's fine. I can take 5 years to implement this idea, but students, they need publications to get to the grad school, to get scholarships and move on with their life, get their jobs. And so I'm trying to create at least 3 different projects. One is kind of that gives them a publication quickly, and the other one very ambitious. If they can get it, it's they can get any kind of job, but at the same time change the field, and something in between those two.
That's a really interesting idea, to do kind of like fast projects versus slow projects. How do you like structure that in such a way? Do you try to make the fast projects build into the slower projects, or is it kind of more of a they're two separate things, and it's more you just got to get the fast projects out for the papers, and then the slow projects where the really like interesting science happens?
I let a person decide what those 3, sometimes 4, or maybe 2 projects are. And for everyone, it's different depending on their unique expertise and their interests. I let it naturally evolve. Don't really have any kind of hardwired thing. It's different for everyone. Something that they're excited about. For me, it's very important that people kind of passionate about something, and of course, it comes and goes in the waves. But I'm kind of, in many cases, just a cheerleader.
Where do you want to take your lab kind of, I guess at the moment you're more short-term focused and just trying to get the grants in, trying to build your lab, trying to get all the equipment, but where do you see it going in kind of 5-10 years?
Well, what we would really like to create is a new nanotechnology that many people can use, that would allow people to create very complex devices in a test tube. So you don't really need to create complicated processes and masks and do lithography, because lithography is kind of very inaccessible. I mean, there are some commercial foundries, but typically you need to have a lot of expertise and you need to confine to those rules that kind of evolved almost similar to nature. And not necessarily, I mentioned that, I want to build a new life form that uses modular design principles, but you look at the current semiconductor industry, it's still somewhat defined by the low resistance pathway. and yeah, so we want to create this DNA, maybe not DNA based, but self-assembly based nanotechnology that people can use to build very complex devices that are not accessible with top-down lithography. And I kind of realized that it will take me much longer than my lifetime to come up with a new life form. And I just want to. I feel our field is already ready to come up with technologies that could be transformational for industry. Maybe not to build a new life form, but to build complex, intelligent devices with arbitrary optical or electronic functions, devices that can reconfigure, that can evolve. Maybe not replicate. I mean, replication is not really, self-replication is not necessary. We just need replications because we can know the structure by measuring it, and then we can just prototype and build many more copies of this structure that works very well. But I want to kind of borrow some of the ideas from biology and directed evolution, but do evolution towards arbitrary function, not something that only accessible to nature, because with synthetic biology, of course, we can already do directed evolution. Nobel Prize to Francis Arnold and others. We can come up with catalytic reactions that far from natural, and I want to take this concept and apply it to devices where we can evolve towards, optimize towards arbitrary function, whether it's photonic, performance, electronic function. And for that, I think molecular programming principles are very powerful because we can build reconfigurable devices.
Do you think DNA is the chemistry that's going to achieve all this, or are there other either macromolecules or other molecular programming chemistries that you think are going to be more of the future?
That's a good question. I think DNA can already do most of it. But having said that, I think there is a room for new DNA-like polymers, because DNA has some limitation when it comes to in vivo or industrial applications like stability and functionality. After all, nature decided to use proteins to build us mostly. Well, many other materials too, but I mean, one of the ideas that I have had for a while is to create a new digital polymer that has capabilities of DNA but overcomes its limitations. And I want to use kind of peptide-like polymer that based on amino acids, but can use this simple principles like AT and GC interactions. But I'm still waiting for a student who is excited about this project. Right now, I don't really have anyone with this expertise. So I have some ideas, but no one really to test it.
Do you anticipate how your group is change over time, or do you have any prediction of that or not?
I don't know what's gonna happen, but I think I'm hoping that we can get some interesting, at least proof of concept studies that would get people and funding agencies to invest more into molecular programming, because I feel like the field has so much potential. And I know there are many other groups who are trying to make DNA nanotechnology industrially practical, and I'm very excited about this. And at the same time, I'm very interested in continuing just curiosity-driven research, like building new life forms. And hopefully, after I deliver some proof of concepts, I get to work on that and get some people who are excited about this kind of things to join my lab.
I know your lab is kind of focusing more on the nanofabrication side and things like that, but do you have any plans to collaborate with like buildercell.org or organizations like that?
Yeah, I actually went to a couple of workshops that Kate Adamal organized, and I think it's also very nice community, and very big community, somehow even bigger, I feel like, than molecular programming. And very early when I started studying chemistry, I was very interested in original life, and I was, my hobby was writing this electron push and reaction mechanisms, or how do you come up with all this purine pyrimidine bases from HCN, and yeah, found a way actually. And when I was in grade 8, not even high school, and then I found this mechanism in the textbook of non-heterocyclic chemistry. But what I feel, yeah, like, as I explained, nature is not very rationally designed. We have this laryngeal nerve, right, that controls our speech, that goes all the way down to the aorta and comes back. uh, so this is such a waste of wiring, from engineering viewpoint. If you could just nip it, and reconnect it on the top, that would make much more sense. But since we evolved from fish, we had to live this with this wiring, wasted wiring. And if you giraffe, this is like 10 feet of wasted wiring. And there are many other examples of kind of very bad design. We have a blind spot in our eye. We eat and breathe through the same passageway, which is a major choking hazard, killing a lot of people. In nanoscale, you have this Rubisco enzyme that is like the most abundant enzyme in the world, because that transforms carbon dioxide into oxygen. But it's like the laziest enzyme possible, because sometimes it picks up oxygen and glucose and converts them to carbon dioxide. It's very inefficient. But nature just took the path of just scaling up the biomass of this protein instead of optimizing it. And nature, whatever works, if it works just better than anything else, it's fine. And so I feel like we can achieve much more flexibility in designing artificial life, building a new type of cell, by letting go the traditional synthetic biology components like enzymes and DNA, and maybe, or using DNA not in traditional sense, but for programming a function in the non-biological function. So if then we can actually find out transistor, resistor, and capacitor that allows us to build very, very complex, but yet modular design that with unlimited complexity. But right now, I don't feel like any community found those building blocks. As you know, synthetic circuits are still quite limited in complexity. And supramolecular chemistry, if you look at the Nobel Prizes, it's way below what DNA nanotech can reach. So I bet that molecular programming will come up eventually with some building blocks that could be used to construct lifelike behaviors. Although I don't feel like it's important to replicate existing life. We just need to come up with something that is as complex, and we can build superhumans that can live longer and better, and have an option of not dying.
Do you think we can achieve that in our lifetimes?
I think we can extend our life enough to with modern medicine to achieve that.
So you're working in the Bay Area where there's all kinds of biotech startups working on both traditional pharmaceuticals as well as, lifespan extension things. Have you gotten a chance to like really interface with that community since you moved to Berkeley, or is this you're kind of like up on the hillside there working on your own little thing?
I haven't really integrated. I mean, I just kind of moved to Bay Area recently, and yes, some people reached out. Some people who run venture capital firms, and they had the ideas on how to do things, not necessarily based on science, and I gave them honest feedback of how I would do that. And there are definitely a lot of people who want to extend their life in the Bay Area. Lots of wealthy people who want to do that. But I'm not directly working on life extension research. I have some ideas on how to create organs and to replace our organs, and maybe one by one as our organs fail based on self-assembly, but now stem cells rather than inorganic building blocks with interesting physics. And they seem to want to fund some very specific ideas like senescent cells, something among the most hottest research lines in molecular biology. And they feel like that if they put enough money into that, that will work. I have a little bit different approach. I explain what the problems are and how I want to solve them, and if they want to invest, they can. But I haven't really talked to many people about that. We're just starting out. But yeah, I'm excited to be in the Bay Area because there are certainly a lot of opportunities to make impact for both industrial, electronics, computer science, and biomedical research. And we just started establishing collaborations where we use DNA nanotechnology to do various in vivo constructs. I feel like it's very easy, sometimes for scientists to maybe overpromise and take people money and they get disappointed. For me, I want to make sure that we have a good underlying technology before we commercialize it, before we ask those people. Bay Area also full of examples of failure when people overpromise and underdeliver. And I don't want to do that. I want to make sure we have a good transistor, resistor, and capacitor before we make a new human out of them.
Thank you so much for joining us, Greg. Stay tuned to our newsletter or Slack for details on our next podcast episodes, and thanks for listening.